The aim of this procedure is to computationally predict three-dimensional structures and biological function of protein molecules starting from their amino acid sequences. This is accomplished by first predicting the secondary structure of the proteins by machine learning. The sequences and the predicted secondary structure are then matched with the solved structures in the PDB library to identify the best possible structure templates. This procedure is called threading. Following the threading procedure, the I-TASSER program will split the templates into fragments based on the sequence template alignments and then reassemble the fragments to full-length models. In the third step, full atomic models are constructed by atomic-level refinements to optimize hydrogen bonding networks and remove steric overlaps. The last step of the procedure is to identify the biological function of the proteins by matching the prediction structures with proteins of known function in the function library.
The main advantage of I-TASSER over existing structure modeling method is the inherent structure fragment assembly approach, which can consistently drive the threading alignments closer to the native state. These high-quality structure models also form the basis of accurate structure-based functional annotations. To promote the usage of I-TASSER in the scientific community, our lab has made available a website where the protein sequences can be submitted to I-TASSER. This website acts as a nexus with which the users worldwide may register an interface to computer cluster which manages and runs I-TASSER simulations.
An I-TASSER simulation job consists of over a dozen smaller sub-simulations. These simulations, when run on a single computer with a single processor core, can take over 100 hours. The Zhang Lab computer cluster takes into